Hi, welcome back. We left off talking about sensation, and for this section, I'd like to start by talking about sensory adaptation. Sensory adaptation is the diminished sensitivity as a result of constant stimulation. So you may have experienced this if you've ever had a cast. When the cast is first put on, you are aware of it constantly. It feels heavy, it might feel tight, it might be irritating you. But after several hours, you notice it less, and by the next day, you're oftentimes hardly aware of it. What's happening is the neurons, as a result of the constant firing, they simply get fatigued. So my question is, what function does sensory adaptation serve? One theory is that by reducing our sensitivity to a constant, unchanging stimulus, we have the freedom to focus on changes in our environment without being distracted by constant, unimportant background stimulation. Do you think that the sensory input to your eyes ever reaches sensory adaptation? Imagine this. Imagine you're driving down a highway in Kansas, for example, where the terrain looks much the same for many, many miles. The road is straight, and there isn't a lot of variation in what you're seeing. If your eyes, because of the constant stimulation, if your eyes reached sensory adaptation, that would be very dangerous, because then those neurons would cease to fire and you would black out. So, our body has a brilliant way of accommodating sensory adaptation in our eyes. Our eyes are always moving just enough to ensure that the retinal image continually changes. Because remember, if our eyes stop moving, the sense receptors would be fatigued, they would reach adaptation, and then the images would vanish. So with that, let's look at vision. We're going to look at the anatomy of the eye, as well as how vision is processed. What is the purpose of the visual system? First, the purpose of the visual system is to transform sensory input that is in the form of light energy or wavelengths into a neural response, which is an electrochemical neural response that can be interpreted and processed by the brain. So remember, in the previous chapter, we learned that neurons communicate both electrically and chemically. In this diagram, you'll see the basic anatomy of the eye. I want to focus on the back of the eye. And this is where you'll see the retina, the fovea, and the optic nerve. Located in the retina, you'll find the receptors for light energy, and these receptor sites are called rods and cones. The rods and cones are responsible for your ability to see colors and see details. But let's tease out the difference between cones and rods. So the cones are the sensory receptors for color and for fine detail. The cones allow us to see in bright light, and importantly, the cones are concentrated in the center of the eye, and this area is called the fovea. The rods are much more densely concentrated around the periphery of the retina, and there are significantly more rods than cones. The rods allow us to see in dim light, but they cannot see detail or color. At the back of the eye, there is a small gap where the optic nerve begins. And it's here that there are no rods and cones, and this is the reason why we have a blind spot. The optic nerve extends from the back of each eye, and it connects through the thalamus. Not all of the nerve crosses to the opposite side of the brain. Some nerve, in fact, stays on the same side of the brain in which the eye is located. But as you recall, that thalamus is the center for directing sensory input to the processing center in the brain. So in this case, the information is sent via the thalamus to the visual cortex, which is located, remember, at the back of the head in the occipital lobe. How do we see color? The cones interpret the differences in the wavelengths of light as color. And because we have 3 different types of cones, this allows us to discern the different colors. The Young-Helmholtz trichromatic theory holds that these color-seeing cones have 3 types of color receptors: red, green, and blue. Furthermore, researchers have found that by mixing these 3 primary colors of red, green, and blue, they're able to create all of the colors that we experience. So for example, when red and green cones are stimulated, we see yellow. Here are some traditional tests for color blindness. Folks who are not able to see the numbers or letters in the center of each of these squares simply lack the functioning red or green sensitive cones, or maybe they lack both. So our televisions are based on the fact that your vision is trichromatic. Just like we have 3 types of cones, red, green, and blue, your TV has only 3 colored phosphors. By varying the proportion of these 3 phosphors, we're able to see many colors. Some phenomenon, however, cannot be explained by the trichromatic theory of color. For example, after images are one example that cannot be explained by that 3-color theory. So what I'd like for you to do right now is pause this screencast and for 30 seconds stare at the white dot that's in the center of the green and black and yellow flag on your screen. All right, after you've been staring at this image for 30 seconds, now I'd like you to look at the blue dot on the right. And what do you see? You'll see a red, white, and blue American flag. So how do we account for this? The opponent process theory states that there are 2 additional color processes. One is responsible for red versus green perception, one for yellow versus blue, plus a third for black versus white. Subsequent research has confirmed that after leaving the receptor cells, visual information is analyzed in terms of the opponent colors red and green, blue and yellow, and black and white. Thus, in the retina and in the thalamus, some neurons are turned on by red but turned off by green. Other neurons are turned on by green but off by red. These opponent processes help explain what you just experienced in an after image. Okay, so in that flag picture, your green receptor cells were turned on so much that they were fatigued or they reached sensory adaptation, so that when we turn away, the opposite cells are firing, enabling us to see red. Now let's examine the sensation of hearing. Sound waves are compressing and expanding air molecules that create stimulus input into your ear. When we look at the anatomy of the ear, the general purpose of that anatomy are to measure the frequency or pitch of sound waves and also to measure the amplitude or loudness of the sound waves. So for the transduction of sounds, the ear engages in the process by transforming these changes in air molecules into an electrical potential in the cells on the fine hairs that are located inside the ear. So because we have two ears, sounds that reach one ear faster than the other ear cause us to localize that sound. So the sound intensity also helps to localize the sound. In other words, more intense sounds are closer. So here's an interesting tidbit. Why does our own voice sound unfamiliar when we hear it from a recording? When we listen to ourselves speak, we hear both the sound conducted by the airwaves to the outer ear, and also that sound carried directly to the auditory nerve via the bone conduction inside our head. So the sound conducted by bone conduction can be experienced, for example, by clicking your teeth. So try that now. The strictly air-conducted sound that other people normally hear, like the sound when we hear our own voice on a recording, that sounds thinner to us because we're used to the combination of bone conduction and air conduction. You can hear the sound waves conducted by bone if you plug your ears and talk in a normal voice. So people who are deaf due to a defect in their inner or middle ear may still be able to hear by bone conduction. And in fact, when Beethoven became deaf, he could still hear a piano being played by placing one end of his walking stick against the piano and gripping the other end in his teeth. So now let's look at some other senses. We have smell, taste, touch, and pain. And we also have two other additional senses. We have a kinesthetic sense, which is the location or awareness of our body. And we also have our vestibular sense, which is our sense of balance. So taste and smell are chemical senses. The taste buds on your tongue are actually receptor sites for 5 different categories of tastes. When food, a chemical, touches the tongue, the receptor sites of either sweet, salty, sour, bitter, or umami are activated. And a signal is sent through the electrochemical process to the brain for processing. Smell is also a chemical sense, but there are many more different types of receptor sites in the nose than there are on the tongue. So here you have an image of the tongue, and those taste buds are showing you the receptor sites for sweet, sour, salty, bitter, or umami. So the olfactory system. Here it's important to note that the olfactory receptor sites in the nerves connect with the olfactory bulb, and this is right next to the brain. Some research has implicated that olfactory bulb is the weak link to the brain, and it even allows pollutants and some viruses to enter the brain through the olfactory bulb. The functioning of the olfactory bulb does decline with age, potentially making us more susceptible. Here you'll see an image of the olfactory bulb. So it's right up there at the top of the nose, and you can see the porous portion of the skull and how those olfactory receptor cells actually penetrate through those openings of the skull into the top of the nasal cavity.